PCR at small microsatellite loci, which I'll talk about in a second, and then viewed on a gel. Pretty standard molecular biology. So microsatellites differ from other areas of the genome. For example, mitochondrial DNA is only maternally inherited, and it does serve to identify variability in populations, but just on the mother's side, for example. So Veronica Conant, I don't know if any of you remember her, she did a senior project, I think, on these salamanders, and she looked at the ND4 gene. She did restriction fragment-like polymorphism in the ND4 gene of the mitochondrial DNA of the same salamanders that I ended up looking at. And she did find some variability. So to confirm her results and then expand on them, Dr. Betancourt and I started working on this microsatellite project. So microsatellite loci are in the nuclear DNA. So they are small repeat lengths of the genome. They show genetic differentiation that's going to be beyond that's just maternally inherited. They're highly polymorphic because their mutation rate is a lot higher. Inherited from both parents instead of just the mom, so they're going to have 2 alleles at every locus. And they're functionally neutral, but because of that high mutation rate, they exhibit some variability. Feel bad. I can tell that I went too far down on my slide because you guys are like, sorry, I didn't think about that. So they're a commonly employed tool in ecological genetics, very commonly employed. The challenge is that their complex mutation patterns sometimes can make it challenging to analyze population structures, which I did find that out. But still, they're very commonly used, and they're easily visualized with PCR and on a gel, which I'll show you a picture of that. So obviously, Scott and Veronica were very important to the foundational research for my work, but Kelly Zamudio at Cornell is like the spotted salamander queen. So she's spent, I don't even know how much time and effort sequencing microsatellites and spotted salamanders. She's also done a lot of ecology studies as well, but I chose microsatellites based on a paper that she published. We just looked at some of the more variable, the ones with high variability in the alleles, tested a few, and eventually found some that amplified well and had some variability. It was mostly the amplifying well that made me pick them. You can use these to look at fine-scale population differentiation in spotted salamanders, which is what she did, so that's what I decided to do. And so I got the sequences specifically out of GenBank and picked just those 4. And AMA is just A maculatum, and then she has a memory system that goes along with it. So there's no real relevance to, the name doesn't really say anything about the locus itself. So for anyone who hasn't seen a gel, here's just a sample gel. Using my pointer. So we mostly used a kind of thicker gel. Usually it was around 2.4% gel, and I ended up using metaphor agarose because some of the base pair, the size differences in these alleles was pretty small, so I needed some good resolution. And then there was a control individual with every gel, so I was sure across populations that I was calling a spade. I was making sure that the allele that I thought was A was actually A across populations. And then a 1 kilobase ladder, at least 2 lanes of every gel, and then we visually analyze the gels to identify different fragment sizes, which are different alleles. So in this one, this is one of the microsatellites, this is another microsatellite. And then there are 2 subsets of a population on here. So these lanes 2 through 5 are Milford, and same here, 10 through 13 are Milford. And then Westford is in 6 and 8, and the same individuals in 14 through 16. So repeated individuals, but at 2 different microsatellites. So normally it was, 1 gel with 1 population and 1 microsatellite, but just for your visual enjoyment, I created one like that. So, got some results, there was significant variation at 3 of the 4 loci that I chose. So that AMA 9.4 was a bit of a bust. Not that much variation, but that can tell a story in of itself. Found 2 to 5 alleles per locus. Let me show you what that looks like. So each color is 1 microsatellite locus, and then the height of the bars is just number of alleles. It goes from 0 to 6. So the most we had was 5, and that was in AMA 33 at the Matches Pond. That's the one at the base of the mountain. So you can see there was some good variability in the size, or in the numbers of alleles. Mean numbers were all between 2.25 and 3.25. There was heterozygosity, meaning different alleles, like one allele was inherited from one parent that was different from the allele inherited from the other parent, and that occurred at all of those microsatellites except for AMA 7. And it was observed that there was less heterozygosity than we would have expected under Hardy-Weinberg conditions. In all the populations, actin was not as distinct. It was probably about neutral. Those were a few of our results. So just a reminder of where we are when I say Milford, Acton, Westford. So Milford's down here. It's 34 kilometers south of these species pools, if that helps at all. 495 is like right here-ish, if that helps anybody. And then again, Acton, Westford, 4.5 kilometers apart. The summit pools are really close to each other, and then the Matches Pond is at the base of the mountains, 1.7 kilometers from the summit pools. Just a reminder.
Yeah.
Yep.
First, do you want us to ask questions during, or would you rather reserve
I
it
don't
to the
mind.
end?
I don't. Whatever you want to do.
So, I don't know. You mentioned 495. Are the freeways a complete barrier to migration for these guys?
Quit asking questions that I'm going to answer.
That's called the segue. Okay.
Thanks for setting me up. I
There you
did knock
go.
it down. My
All
fault. I will talk about that later.
Cool.
It's disputed, but yes is
Okay.
the short answer. So I just took kind of that map and then I overlaid the allele frequencies on it. So each of these circles represents 1 microsatellite, and then the variations of gray are the different alleles in the pie, in each pie, and then the size of the pie is correlated to the frequency of that individual allele. So if that makes any sense. So you can kind of get an idea of like how geographically these are distributing, the allele frequencies are distributing. So just for an example, if you look at this microsatellite in each population, those are pretty similar, pretty close to each other. Very different from here. The most common allele here is almost totally lost in this population. And then on the summit, you can see kind of similar allele frequencies. There's an allele here that doesn't even exist in the other summit population. And then at the Matches Pond, there's way more variability in the number of alleles and the frequencies than the summit mountain populations. So just as an example. So these are the same pie charts, but just on maybe an easier to see format. The gray boxes just represent geographic proximity to each other. So this is Milford, it's kind of on its own, Westford and Acton, and then the mountain populations, and then the numbers at the top are elevation. So Matches is at 390 meters elevation. The summit populations are around 600. The lowland populations, as I call them, are Westford and Acton and Milford. Okay. So if we did some statistical analyses of these frequencies, we found that across all of the loci that we tested, there was similarities between Acton and Westford, which you might expect, and similarities between the summit populations, and that was based on fixation index, or FST, and Nye's genetic distance, which thinks a little bit more about drift influence. And then Matches is different from the summit. So you can see these pairwise values are just looking at how similar, based on FST values, is the upper summit to the lower summit. And 0 is like exactly the same pretty much, and 1 is completely